All right. Welcome, everybody, to the second of our Response Diversity Networks seminar series. And I'm Mike Fowler. I'm one of the steering group members, and I'm very happy to welcome uh, Jeff Arnoldi to give the second seminar. Jeff started off as a physicist, I think, and developed an interested in studying the dynamics of chaotic systems before going on to work with Michel Legault in SETE in the Theoretical and Experimental Ecology Station in the south of France. It's a very beautiful place. And he went on to do a postdoc with Andrew Jackson and Ian Donoghue in Trinity in Dublin. And now he's got a permanent position back at SETI, um, working more on theoretical ecology. And so his work, I think it links very neatly and very directly to response diversity. And he said he's going to take this maybe in a slightly tangential direction, but I think it's still going to be of interest to everyone here. And his work deals typically with multi-species community and communities and how they respond to environmental changes across biological and spatial and temporal scales. And he's explicitly linking classical theory with empirical systems. And his work considers the emergent phenomena from these complex ecosystems to reveal underlying structures and patterns in their in these ecosystems' dynamical responses. So he's dipping into functional ecology today, and I'm really looking forward to this. So with that, I'll just pass on to you, Jeff, and let you take over. Thanks.
Hey, thanks, Mike. Okay, so today I'm going to talk about functions, ecosystem functions, and how species contribute to those. And in particular, I want to try to take it as far as I can using simple mathematics, the linear algebra, that can still contain some interesting complexity, I think. So this idea that there are functions, it's an old notion in ecology. So functions are typically processes like respiration or nutrient cycling that are to which there's like species participate to collectively, and it understanding how different species contribute to that has some obvious implications for conservation, for instance, like trying to figure out who are those very important species for the certain processes that are key to the livelihood of organisms and also humans. It can also relate to applied notions like trying to figure out what are good productive mixtures, for instance. But the starting point of my talk will be a very simple, let's say, um, framework to think about species contribution, which is the mass ratio hypothesis. And there's this notion of the way that we relate species to function is through a notion of effect rate and abundance. So this idea is that the way a given species is going to contribute to an ecosystem function relate to the traits weighted by the abundance. And so that's literally how people scale up from organisms. So traits as things that you might hope to find on organisms, so organism characteristics, and then you scale up from organisms to a community and ecosystem by just by weighting by abundance. So that will be my simple starting hypothesis, and I'm gonna use it and run with it as far as I can. So what this mass ratio hypothesis of Brian basically says is that functions are linear in abundance, so biomass. I'm going to use abundance and biomass a bit all over the place. I'm sorry if that's offensive sometimes. So the idea is that you have in a given ecosystem that's in a given state, you have some species has a certain abundance, a certain effect rate that basically is the way you transferred like from biomass to the function in question, and you just add those contributions up, weighting by the abundance to get the whole function in the end. And okay, so another way to say this is that it amounts to viewing functions as directions. So what do I mean by this? If I take total biomass, I have a given ecosystem that's in a given state. Here, two species for representation. The total biomass is the sum of the biomass. Okay. But another way to view this is to say that I'm weighing the effect rate in this case for total biomass is just one. You just attribute 1 unit of biomass per unit of biomass, obviously. But when you write it like this, you see that it's like a scalar product between the state vector, which is the joint abundances of the species, and a vector made of ones. And when you do this scalar product, you just project it like this. And if you had another state, you would project it, it would be somewhere else and it would be a different total biomass. But the way you would get total biomass is to make the scalar product with the same vector. If you had another function, maybe that gives more weight to the first species here, for instance, you would also have this single direction and different states would project on it. And if you're thinking of different functions of the same ecosystem, well, you're basically projecting on different directions. And that's the idea. So it means that when you're thinking of functions in this way, from this perspective of the mass ratio hypothesis, you're thinking of directions in some state space. The state space has axes that are the biomasses of all the different species, so it can be high dimensional. But those functions are directions in there, and it begs the question, is there some geometrical structure to multifunctional ecology? And this is the first part of my talk is going to start from there. And it's a way so it was led by James Orr, who did his PhD in Dublin, and it was published only recently. But the way I'm going to present it here is really to say, given this mass ratio hypothesis, it implies certain things about that could be interpreted geometrically about systems. And so we can test this hypothesis and see how far it goes by pushing it into this direction, this geometrical direction. That's what we did. And what we did was to revisit some meta-analysis of impact factors on soil microbial diversity and function. So it's basically soil microbiomes and various functions here, like biomass, like microbial biomass, respiration, some other functions related to carbon, phosphorus, or nitrogen cycling. And importantly, what is being reported is the responses of all these functions to various classes of perturbations like warming, pollution, land use change, and so on. And what the kind of data that we analyzed is what we call mismatch data. So it's this idea that you're going to look at two different functions and look at their response to a given perturbation and see if they are responding qualitatively in the same way. Is one increasing when the other increase, and vice versa. And what we tend to see in this kind of data is some mess. It's quite messy. A lot of time there's a fair amount of mismatch. For instance, if we look at the biomass on the left, it's biomass and respiration. You have a large fraction of mismatches, but it's even worse when you look at measures of diversity and some measure of functioning, where you have like more than half of the time it's going the opposite direction. So it's already pointing to something a bit odd that's going on, where diversity and functioning are going in opposite ways, which is never something we like to talk about, but we'll see where it's coming from. So if I organize the data a bit more systematically, this is what it looks like. So we have those, here we have relations between diversity and functions. When we see a lot of red, so this is where like we see the diversity and functioning are going in opposite direction here. We see here, for instance, that if we focus on hydraulic carbon cycling, there's a lot of blue, so it means that it tends to be coherent for functions that are related to similar underlying processes, which makes sense, obviously. And then we have between certain functions like respiration and this hydraulic sea cycling group of functions, like something that looks way more random, where there doesn't seem to be much of a pattern there. Okay, so let's try to see if we can make sense of that somehow. And the way I want to make sense of that is to use this notion of functions as directions. So here we have a representation, artificial representation of two functions, respiration and microbial biomass, seen as two directions that make an angle between one another. And to relate that to the kind of data we have, what we can do is say, okay, I'm going to position myself, I'm going to see perturbation as a change of state, of my ecosystem state. And I'm going to put it at the origin. Because functions are linear, I can just draw them again as direction that goes through this. And it's not function anymore, it's function response. It's how the response, the function changes starting from this state. And so if I do a perturbation, whatever it may be, it just changes my state. And then I do this projection back on my directions that represent the functions. So here in this artificial example, when I project them back, I have a negative response for both. So it's consistent, it's a coherent response of both. But if I had perturbed my system here, where I just put it, biomass increases, but respiration declines. So that's where that would be a mismatch. So where is there a mismatch? Geometrically, there's a mismatch because we're falling into this cone that I've drawn just by drawing the orthogonal lines of both directions. And the angle that this cone makes is precisely the same angle as the two directions make with one another. So if this means that if my perturbations were just randomly scattered all over the place, the fraction of mismatches that I would get would be theta, the angle over pi. So if I go back to my idea, the notion that I have this linear view of functions should means that there's a geometric underlying structure, and so explaining mismatches would amount to a measure, in this view, would measure the angle between two directions from knowledge of the angles with others. So if, for instance, in this very simple example, you know, the angle that is unknown here can be deduced from the angles that the other is just the difference. But you can extend this problem in high dimensions. It's a linear algebra problem. It's not a crazy hard problem. But what's important is that it tells you how you should predict things from the data. And it's not like, you know, I don't want to do like a statistical, let's say, correlation between parts of the data with another. No, I really want to predict mismatches. So a fraction of mismatch I see between two functions from the rest in a very specific way. And if I take the data that we have and I just pull together all the biomes that were in meta-analysis, what I get is nonsense. It doesn't predict anything. And that makes sense because here for this geometrical view to hold, I need to think that there is a given underlying phase space, like a space state where the axes are fixed. So it means that the species are fixed more or less, and the functions are fixed as well. But if I'm pooling too many different ecosystems together, it doesn't. Uh, I could still have a linear characterization that would make sense, but because I'm considering systems that are too different, it's no longer correct to. I cannot really make the interpretation work. But when I select for just grassland systems, then I get something much better, much more convincing. So I should be close to the 1-1 line, and this is what I see. So this means that this mismatch data that I have can be translated as angles between functions, and angles between functions in this sense can be thought of as distances between them. So how dissimilar they are from one another. And when you do this, you can make a network of functions and organize them based on how distant they are. When we do this, we recover something relatively coherent, where we group together the functions that are mechanistically related to one another. And another interpretation you can give to this is how broad the function is, because broad means how evenly all the different species contribute to the function. And the most even would be just total biomass, because everyone contributes exactly the same per unit of biomass to biomass, obviously. And so the angle that you make with biomass, it can be a measure of broadness. And when we do this, we find that the broadest function is respiration, as we were expecting, and then some mineralization, and then like phosphorus and like some nutrient cycling functions appear then. Okay, but one thing I want to really focus on now is this idea, this a bit uncomfortable realization that there's a negative diversity function relationship somehow in this data. It's quite systematic that functions and diversity go in opposite directions. So how I want to try to give an interpretation of why, an explanation of why, based on geometry. So now when I'm going to talk about diversity, the best way is to think of it in a general sense, like, for instance, Hill's measure of diversity. So it's more a measure of evenness in the community, with species richness being really uh, an extreme case. But so now what we can position diversity in the same way as we did with function, except what we're going to put is the gradient of diversity, how diversity changes when I change the abundance of a certain species. And so the way you have to see this is to say the axes are not organized in any ways now. I have to think of who is the least abundant species, who is the most, if I have just two, for instance. And because when I increase the abundance of the least abundant, that increases diversity. Whereas whereas it's the opposite for the most abundant, increasing it makes diversity decrease. That's why diversity is now pointing a bit downwards. It points downwards, fine, but it's never going to point downwards enough for the angle to be larger than pi over 2, which would be needed to explain this idea that there's really systematically a mismatch. It's not just half and half. It's like really like sometimes it goes up to close to 70% of the case where diversity and function goes in opposite directions. So that doesn't explain it exactly, but there's a way to explain it. But if we understand that the perturbations, they act somehow, they must scale somehow, or it's reasonable to expect that they scale with the actual biomass of the species that are being perturbed, meaning that species that represent a lot more biomass are probably going to change more in biomass in absolute terms than less abundant ones. And when you do this, you see that the cloud of points here that represent perturbations should be stretched upwards. And this stretch upwards can be thought of as a rescaling of the axes. And this rescaling of the axes, what it does is that it forces even more the differences that existed between the directions made by the function and diversity. And now you can very easily get an angle between function and diversity that exceeds pi over 2, which implies like systematic mismatch of the kind that we see in the data. Okay, so the point of this first part, and there are many other things to be said about this work, but the point I really want to stress here is that this idea of the mass ratio hypothesis, it makes sense. It has quite a bit of merit, even when we think of complex functions and complex ecosystems like the ones I showed. Like it works quite well, I find, in even a bit surprisingly so, probably. And this geometrical structure of seeing functions as direction can reveal features of functions and perturbation. And something that would have been nice for me to show if I had more time is that it can tell you something about the response diversity depending on the class of perturbations that you're going to have, and we see that different types of perturbation show different kinds of levels of response diversity. So it reveals features of functions. It can reveal features of perturbations. But this mass ratio hypothesis, by definition, attributes most of the functional role to a few abundant species, because typically communities are not super even. There's like a few very dominant species in there, and it's precisely because of that we see this negative diversity function observation, because the way an actual perturbation is going to change the biomass, the easiest way is to remove the biomass from the most abundant, and that increases diversity. So that's an up and vice versa. So this idea that there's abundant species that do a lot of the work and that there's a negative diversity function relationship is like two sides of the same coin. But why is that? And why don't it's hard to really settle with this idea, is that it's because all of this is a static view. It really looks at the ecosystem in a given state, and so it actually bypasses ecological dynamics. It doesn't account, for instance, for the idea that there could be a keystone species that indirectly really maintain together the system, or like, for instance, pioneer species that have, through the colonization of the environment, allowed for new species to come in and actually have some function today that, well, whereas they are fundamental for what we see today, but then might not even be here anymore. But these are keystone species, pioneer species. Everyone has those examples in mind. But we think of them as historical, context-dependent cases, and also very nonlinear. A lot of the time, we invoke complex scenarios to explain this idea, and probably these complex scenarios occur a lot. But a point of what I'm going to try to make now is that it's a lot more common than that. It's generically there's going to be a notion in which species that don't look important from the mass ratio hypothesis actually are. And that's the topic of this second part. When we try to explicitly account for ecological dynamics, how will that change our perspectives on contribution to function? So I'm not refuting the mass ratio hypothesis. I've just showed you that it made a lot of sense. I'm just saying its actual interpretation has to be taken with caution. So this is ongoing work by Alice Ardishvili, who's doing a postdoc with me, and Michel Laurent. All right, so the idea is that we had, and I'm still going to start this as my premise, this mass ratio hypothesis is like a linear, a static linear partitioning of a given function. So we just we have whatever function you want, you just sum contributions, and contributions are related to abundance. But what I want now is to move away from that and define a dynamical linear partitioning. And this linear partitioning, I want to reveal it through perturbations, because perturbations reveal dynamics. So here it's conceptually, I want to perturb a given species and see how that affects the function. So there's perturbation and response. And this response, I want to let it be over long, like over ecological timescales. Ideally, I would like to have a theory that accounts for various timescales, but here you have to think long ecological timescales. So how am I going to do this? Well, I just do this as I know how to do it. Just do derivatives. So I'm going to think of the perturbation as a change in some demographic parameter. Let's call it K, Ki of my species I. And I'm going to see how the function response is sensitive to this variation. So I'm just going to do the partial derivative of the function with respect to Ki. And this when I write this like that, I have two terms. And the first term that I've framed has the units of the function. And the second term, I have to, it has no dimensions, and you have to think of it as like a relative measure of the intensity of the perturbation that I made. And this part that I framed, I'm going to call it phi I din, like for dynamic. And as I'm going to show that this is going to give me the dynamical contribution of the species in a specific sense. Okay, so let's work our way a bit through it. What does this mean concretely? Well, first of all, I've introduced an artificial notion here, K star, which is you have to think of it as abiotic conditions as are perceived by that species. So it's you could think of it as the species monoculture abundance, for instance. And if I develop a bit my derivative, what I see is that if I plug in the mass ratio hypothesis, so I'm really building on this, I'm going to have an emergence of different terms that show the effect rate of all species and their interactions between them, net interactions. And if I can define my weights, the K stars, so that's like a key little trick. The K stars can be defined in a certain way so that for any function, when I sum my contributions right in, the sum gives me the actual function, exactly the same way as we had for the static partitioning. So we have an additive partitioning. So we went from this static partitioning that goes abundance to effect rate of the given species to a dynamic one that accounts for interactions. And so this is a bit how it goes. You look at a given species, you see so you have to read this from right to left. So you look at there's a given weight given to the species K star, and then this species connects through net interaction to all the other species and their effect rates. And so that's the dynamical contribution of the first species. And you add to that, you add the same for the second, the third, the fourth, the fifth, and the whole thing adds up to the overall function of the ecosystem. Right. So we can go a bit deeper and get some insights into what goes on in here. The idea, and I want to do this because I want to connect with some notion of that were quite inspiring for me of work by Alvaro Sanchez in microbial biology, where they find that there's a notion of background functioning that accounts for a lot of the context dependency of the effect of inoculation by a strain of bacteria. But maybe I'm going a bit ahead of myself. But what I want to do here is try to break it down a little bit, saying, okay, let's pick one species, and for convenience, I'm going to call it species 0 to isolate it from the rest. So I can write the dynamical contribution of this species as two terms. The first part is things relating to the species, so with index 0. And the first part that I've squared, the partial derivative of its abundance with K0 is a bit tricky, but it's like a sensitivity of that species to change in abiotic conditions. And then the second part has to do with the other species from the community. And here I'm going to do the cavity trick. So it's like remove and add that species. So it's like I want to consider the community without that. I artificially remove the species and I put it back. And the why I want to do this is because I want to understand what would have how the abundance of a given species in the community how sensitive it is to that particular species that I chose, species zero. So the idea is that I want to say, okay, my species in the community, where that species zero is the abundance that it was when that species was not there, plus a term that is like a response to a perturbation, like the invasion of this zeroth species is like an environmental perturbation that is probing the dynamic of the resident community. And why I want to do this is because if I go back to the expression of the dynamical contribution of species, I want to see how the abundance of others are sensitive to change in abiotic condition of my focal species. So the net interaction between it and the others. And by doing this trick, I've took out terms that cannot depend on it anymore. So when I will take partial derivatives, it will just do nothing. And now, so this is just like, I mean, if you're watching this somehow, you can just pause and do the calculation yourself. But what happens now is that if you replug that in, you see appearing the dynamical contributions to the function of the resident species in the resident community, so without the focal species. So the point of that is that if I write that all back together, I have this prediction that my dynamical contribution is the sum of two parts that are quite intuitive in a way. So we can skip the first line. That's the exact term, and the second one is like more of a way it could be made. It can be exact or interpreted as an approximation, depending on what sense you put on the A bar. But the point is that you have a first term that is the functioning of a monoculture of that given species. And then the second part is background functioning. So the function in the absence of that species times the interaction, the mean or effective interaction that this species has on the others. And this is precisely the kind of things that Alvaro Sanchez and his team have explored in bacterial systems. So I'll explain it a bit later, but the idea is that a lot in the work of Alvaro, a lot of the context dependency of the effect of a species in a community can be really accounted for in this background functioning term. All right. Intuitively, this notion that a species has a given dynamical contribution to the function relates to what would happen to the community if that species was not there. So we can do this conceptually, remove the species, look at the function without it, and define a net effect. Define in a way that if positive, it's like if the species had a positive contribution. And if we do the computation, well, we recognize again this dynamical contribution explicitly. Like basically the net effect of removing a species is proportional to this dynamical contribution. Why is that? Well, because removing the species, it's a bit like if there was somehow a targeted press perturbation focused on that species and pushing it all the way to extinction. And how strong that perturbation needs to be depends on how resistant the species is. And so that's why there's this second term. That's the proportionality term. But it's a nice term in a way because it has a notion of N over K, which is relative yield. So that's a measure, something that can be measured. And it has this V naught term that is a bit more um, mysterious, but you can think of it as being close to one. And so obviously, like it will have more of an impact if to remove a very resistant species or a species that is like super abundant. So it would take a lot of it. It's removing it amounts to doing a big perturbation. But the point is that removing a species is proportional to this dynamical contribution. Okay, so now to the real stuff. So here I want to revisit some work done by, I'm super glad some of the team that did this work are listening to me. I'm glad and also a bit afraid. So it's this work on ciliate communities. And what I'm going to do here is do this thing of assembling communities and looking at what happens on. So the function I'm going to consider for this empirical case study is total biomass, the simplest thing I can do, for which the mass ratio hypothesis is obviously correct, like the sum of individual biomass is total biomass by definition. And I can do remove add of species of this. So I have the 5 species. I know that in we removed the tetrahymena because it was going to extinction all the time. Right. And so we did this little experiment. And what I want to show you here is organized in like 2 levels. First, I want to see when I remove a given species, is that if is the effect basically accounted for by the mass that I've removed? Or in fact, the way it's defined, you can think of it more the other way around, like of how much mass if I add the species in, it adds some mass. Is this all that's going to change in community biomass, or is there going to be differences? We expect differences because typically if a species has a lot of biomass, it often comes at the cost of the biomass of others. But as we will see, it's not always the case. And then my second hypothesis has to do with the theory that I presented, where I'm going to use, and I won't explain that so much, but I'm going to infer somehow the net interaction between species to have a prediction on the dynamical contribution and the resistance of the species to predict what would happen in case of the addition or removal of a given species. So this is the first part, the first hypothesis. So this is just on, so the y-axis won't change in the next 2 slides. So that's how much biomass has changed when I add a given species. And here I compare that to how much mass I've actually added. So when I see when the effect is negative, it means that somehow total biomass decreased by the addition of the species. And so we see that for a few species, it can be negative. We see a pretty poor correlation between mass and change. We see some, if you look on your right, you have an estimation of the pairwise interactions between the different species. And you see that it's consistent, like facilitative species tend to be above the 1-1 line and competitive ones under the 1-1 line. And okay, and if I move to the prediction, then I get something a lot better, which predicts relatively well the effect of removing or adding species. And see, so here what we did is that we inferred those dynamical contributions to then predict through this. Like we basically used the idea was to use introduction of a species as a perturbation that probes the dynamical contribution within the community, and do that to have an estimation of the dynamical contribution of everyone, and then predict what would happen if we removed each of them. So I'm guessing what I just said made no sense to no one, but that's fine. What's very important to me is this figure is that if we compare dynamical and static contribution, so static contribution here just means the mass of the species here it's relative mass, and the dynamical contribution, we find no correlation. It's not significant at all. And what does this mean? It means that the fact that a species is important for the given function is not really related to how much it actually contributes in the given state. Here, like how much mass it has, how much important a given species is mass, how important a species is for the overall biomass of the community is not really related to how much biomass it actually has. And for instance, we have some funny things. For instance, this one has always very positive, but contributions, yet its interactions with the rest tend to be negative, weakly negative. And this one, we see another strange pattern as well. And here, I want to see this. There's this idea that, so here there's this notion that it's a bit all over the place, but I can understand things a little bit better when I organize them via the Sanchez view of comparing the dynamical contribution with respect to the background functioning. So here, the different colors are the different species, and the different points are different communities in which the species is. And those different communities have different background functioning, so different overall biomass. And we see, for instance, that for Paramecium, we have a pretty clean linear relationship between the two, and it's a negative relationship, which means that it tends to the indirect effect of Paramecium is negative. But for the green, in the green, we see something much more positive because it's much more of a facilitative species. And as predicted by Sanchez's work, a lot of the context dependency in a dynamic contribution is accounted for by this background functioning. And so that's the results of some inference that you can trust or not on the data. It's a bit tricky and technical to explain concretely what we did, but you can do the same thing with simulations, where you can test that the theory is exact, and the kind of patterns you see in the end between this relationship between dynamical contribution and static contribution is the same. There's no correlation, and you can have mismatches between what you think was like positive contribution and direct interactions, what they really are, and so on. And so to conclude, there's like two ways of relating species to function. There's this static and dynamical one, and the static one relies a lot on effect rates, and it holds this promise that you can look at individual organism level property, and that are called traits typically that you could measure and scale up nicely. But the problem is that it may not accurately predict response to perturbations, because response to perturbations probe dynamics, and this view is static. It says nothing of the ecological dynamic. The dynamical partitioning, on the other hand, is much more collective and can be context dependent. we saw that some of the context dependency can be accounted for somehow, but it's still unclear exactly how. It's difficult to observe, but it can be inferred from perturbation. So you have to use perturbation to probe the underlying structure of the system. But most importantly, this dynamical partitioning is defined to predict the responses to perturbations. And if we think of what role is given to rare species, for instance, there's a contrast between the two perspectives, because when you take the static perspective, a rare species can only be important if it contributes to a very narrow function. So if it has very unique traits that relate to a very particular function. And that's trying to figure out which species are functionally important in that way is like the focus of a lot of work. But if we take the dynamic perspective, rare species can be as important as dynamical ones. We saw no relationship, in fact, either in the data or in simulation between dynamical and static contributions. And that's because it's a generic outcome of interactions. It just complexifies the network enough that the biomass of a given species is a collective outcome. It doesn't reflect its intrinsic properties so much. And that's how you can have, and it's been established also empirically in like grassland systems also that a cohort of rare species, if you remove it, it has a disproportionate impact on the overall yield. Um, there's also work by Dauphren that shows that large ungulates, even though they don't represent much biomass in the forest, they are key to nutrient cycling through stoichiometry. Yes, that's it for now. Thanks.
Awesome. Thanks a lot, Jeff. I hope that recorded. I really hope that recorded properly. But it was a great presentation. I always enjoy um, listening to you talk or reading your papers because they always provoke some kind of interesting thoughts in my own mind about ways to dig into these questions. And then it takes me years and years and years to get my head around it. So I'll maybe come back in a couple of years with some questions about this. But if anyone else has got questions just now, please use the hand raised reaction button.
Was that a nice way to say that just confused you?
Uh, I wouldn't. I honestly wouldn't put it like that. I think there's my brain just doesn't work as quickly
It's
as
fine.
yours does, maybe. So, um, Pete, you've got a question, please. Do you want to unmute yourself and go ahead?
Yes, yeah, thanks a lot for the talk. Very interesting, and learned a lot about some things I didn't know about. I had this the question, about your final conclusion was are traits still useful in this dynamical approach? And then I was thinking about what you termed like background functioning, and I wondered about that background functioning is obviously a combination of the current environmental conditions and the current species composition. And then a new species entering that system can either can enter or can't enter the system based on those environmental conditions and the current composition. And its traits are also different to they can be different to the community it's invading in or leaving in a variety of different ways. And so I wondered, like, when you take that kind of perspective of like, where is background functionally, where is the environmental conditions functionally, and what are the traits of the invasive species entering relative to the species that are already there? Does that still, a species still a trait still useless when you take that approach, or uh, I'm not sure if I'm making sense here, but I'm just kind of
No,
thinking.
I think the way I understand it is that if you take, so maybe it would be a complex answer. If you take this expression, so that's That's the formal expression of the dynamical contribution. So it's just a linear theory on what happens if you perturb a species and how it's going to affect a given function. You see the effect rate of everyone, you see the interaction with everybody. So it looks like it really diffuse the very collective how it goes and hard to relate to the characteristic of the particular function that I'm looking at, right? But through this trick here, what you end up finding is that, well, there is a way to simplify a bit all of that by having background functioning as a term in there that encodes for a lot of the context dependency and the traits of the species present in the community, so that what you are left with to understand what is this particular species that is doing, I have it's functioning in monoculture, so that's really its traits, functional traits. You have the background functioning, so that's really a property of the community it's going to be put in. And then you have this second term that is how the kind of interaction this species has with the community. And so there, it's it could have been just a way to rewrite things that wouldn't really help, but somehow it turns out, and I don't understand well why exactly at that point, but that's Sanchez's work shows that a lot, that this background functioning explains a lot of the variation in a way. That you can say, for instance, oh, a given species is a facilitator. So it has this A bar is kind of positive across the board. And how that is going to manifest in different communities is going to be explained by the background functioning. Right. And so that's the point of this figure. You see, like you have the lines. Well, of course, if the line is flat, then the linear relation doesn't explain anything. But for the green one, for instance, you have a fair amount of variation that is explained by the linear regression, which is another way to say that a lot of the context dependencies are accounted for by background functioning. So yeah, I don't know if I don't have a complete answer. I don't know how to relate.
Yeah. I guess my comments a little bit like just to extend what you were presenting is that that interaction between the background functioning and the species which you've got represented by its monoculture performance, that somehow that is a kind of trait dependent process itself, and that the kind of traits of the community interacting with the traits of the entering species that like determines its impact in a way. It's like,
Yeah,
where does
yeah,
it sit relative to the ones that are already there? That's maybe is a sort of trait predictable functioning,
yeah.
or
Well,
trait predictable. If you could understand those rules, you could understand how species impact a system when they leave it or enter it.
hmm. Yeah, well, there's the VI term is very important. That's has
Mhm.
something to do with the feedback that the species experiences from the community. I've left it out a bit here, but I've done work in the past specifically on this kind of term.
Mhm.
And that, yeah, I don't know. At this point, I'm not really, yeah. My conclusions are some are positive, some negative. I don't know. I don't know. Depends on the moment.
Yeah. I guess I think I guess my sort of idea is that I think you can that is a term that it does have a biological basis,
Hmm.
and it's something to do with what the trait array of what's already there is, and what the trait of the new
Oh,
species
yeah,
is.
yeah.
Yeah. Hmm.
Yeah, most
Yeah.
likely. Hmm.
Hmm. Thank you.
Awesome. Thanks. Owen, you or someone in your group are free to unmute and ask a question.
Thanks very much, Mike. Thanks, Jeff, for the presentation. It's lovely to see that data being used. I'm sure Frank agrees as well. In when one's thinking about response diversity, we often use monoculture information from monocultures, or like fundamental niche type of responses. We take we give each species or measure each species response, which would be a response trait, and then maybe even weight it by abundance or biomass, and then calculate some summary statistic of the distribution of those responses, response traits or weighted response traits. And then often the question is, what about interactions? When the environment changes and when the abundance changes, and I wasn't totally able to see the connection here, if there is one, between what you explained here and that question of how interspecific interactions could affect that kind of measurement of response diversity. Can you help with that at all?
Yeah, of course. Let me think of the good slide to show. Maybe it's really in a way it really starts from the definition of the dynamic partitioning, which is really it's saying what here I said what happens if I do a perturbation on one given species, so a change in its carrying capacity somehow. For instance, it could be via a temperature change that we see that in the monoculture it changes its abundance, so that can be a measure of the. In fact, I'm analyzing your data again with that recently on a different topic, but similar. So here to define the dynamical contribution of one particular species, I imagine a perturbation that would affect just that species. But in fact, once I have this definition, I can revisit it by saying what happens to it if it had been a general perturbation that affected everybody. Then what you would do is you would just sum. You would weigh all the different perturbations that could be potentially measured in monocultures. You would weigh them by the dynamical contribution, and you sum that. And that's how you would predict the overall ecosystem response. I guess it's a bit hard to explain in words when it would be very easy just to write, but I think the point here is that this accounts for interactions, and it would predict an overall response of the ecosystem to a more general perturbation that would affect everyone. Was that an answer, or maybe not? Sorry.
Yeah, thanks. Maybe we'll come back to you some other time and ask you to write it out for us as well. Thanks.
Okay.
Yeah, I'll go to Adam next, and then I might add something about Owen's point in your response, which I thought covered a question I was thinking of as well. So yeah, Adam, take it away, please.
Feel free to go first.
No, go ahead. No, I don't want to hog the limelight here. Go ahead.
Well, nice talk, and good to see you again, Jeff. I think this is a very cool topic, and is maybe the closest I've come to understanding the cavity method so far. So I appreciate that. This is a bit, this may be a very stupid question, in which case I will blame the fact that I probably have a 39-degree fever right now. But this feels like, among other things, you sort of derived a more sensible null model for a lot of the BEF stuff that we've been doing. And that, like, the standard null model is that you assume you're comparing species yields in monoculture to their expected yield in mixture. And the expected yield in mixture is usually monoculture divided by the number of species, which I think all of us know is it's a statistically useful value, but it's not what species do in the real world.
Uh-huh.
And it feels like what you're doing here through this dynamical version is, among other things, coming up with a more sensible sort of null expectation for what you would expect a species to be doing in mixture, based on some kind of an underlying dynamic model. But maybe I'm being crazy.
I don't know why you say no. Yes, yes and no. I mean, it is a theory for how uh, what would happen if you add species in a given system. So it is basically okay. I can answer this indirectly a bit, but why did I even start working on this? I started working on this because I started to feel that there was a weird um I don't know, like not confusion, but like some sort of a contradiction in what I could hear people say, depending on what background they had. Like if they were like coming from functional ecology or BEF, like BF, like biodiversity ecosystem functioning, people say, oh well, more and more species, higher diversity, more function. Okay, fine. And then if you hear people that talk about community weighted means and traits, they would say, well, actually a lot of the functioning is accounted for by like 80% of the variance in function is just a few species. So it's like really, so it gave me this feeling that there was an awkward tension between what role you're going to give to diversity itself. Like on one hand, you say it has no role, the other is saying it has all the role, and yet you're both describing the same systems and accurately. So and so yes, and so that's why for me it was important to say, well, if I look at the overall biomass of a community, so that's the simplest way. I have an additive partitioning. I just add the biomass, and yes, it's true that in many cases, maybe 80% of the biomass is just two species out of 100. I don't know, maybe not 100, but whatever. And but is it true though that a lot of these rare ones have no role in this? Given that the biomass, in fact, and you feel that it probably not the case because if there are interactions between species, if you have dynamics, the biomass of a given dominant species is not an individual trait; it's a collective outcome. So that's why, and this partitioning tries to show that you have to the way you have to add things up is different. So in that sense, for sure, I don't know. I wouldn't call it a null model because a null model would I don't really know. Yeah, I mean you can always say that a theory is always going to be a null model, I guess. But
Oh, so I wasn't thinking of this as a null model, but was rather thinking like if you can take the simplest type of interaction model that you're willing to imagine for your system
yeah,
and then test that against a more carefully
right.
parameterized one and have
So for me, the notion of the null model in BF is more like a way to formalize a very basic intuitive expectation, which would be that actually the overall like, think that it's a zero-sum game. Like you say, it's a zero-sum game. That's all there is. That's my null hypothesis, and so that's the that's on yeah the net biodiversity effect is like a deviation from this zero-sum game hypothesis. So here I'm not making this exactly, um, but yeah, for sure it's I would like to make it a bit more explicit how that directly connects to um, biodiversity ecosystem functioning, like how what happens when you keep adding species. And I think the answer lies a bit in again in this expression here, this last expression, like you're going to add a species, it's going to depend on the context in which you're adding it, the interaction it has with others, and that's how it's going to build, yeah, incrementally.
well, thank you. I think I may send you a follow-up email.
Thanks for the question.
Yeah, thanks, Adam. Last chance for anyone else to jump in if you've got questions. While I'm waiting for any last hands being raised, I might try and ask, or maybe repeat or rephrase part of your discussion with Owen, which I was thinking about during your talk as well, which is the sort of perturbation that you apply that can only affect a single species, which you then, in your response to Owen, you covered one way of dealing with that, which is, well, you can apply a perturbation that affects everybody's K naught or Ki star value. And I think actually you can come up with perturbations that should emerge from whatever mechanistic model you're using to or mechanistic framework you're using to come up with your dynamical model. So that the covariance structure of the way that the K star values respond to a single perturbation across an environmental gradient or a nutrient gradient or something like that. I think you can make the link to response diversity through that mechanistic framework. Is it a reduction in all nutrients? Is your perturbation a reduction in all nutrients across that concentration gradient, maybe in an equal way, or have you got an asymmetric change to the nutrient concentrations because of the sort of perturbation that's applied? And then how does that affect the K naught values or the Ki star values? That's kind of going at it from a different direction than I think you've gone at it from, which is just take a single Ki value and then what do you do? Reset it to 0? Is that right, or something else?
Sorry, the reset what to 0?
The Ki star value. Maybe you don't reset it to 0, but you change it. You change this.
Yeah. And so the idea, if you want, is that so again, it would be easy if I could write, but you have the dynamical contributions. They are the underlying dynamical structure of the community, if you want to think about it. And then the perturbation is going to probe this.
Yeah.
Going to ignite this dynamical structure. And depending on the perturbation, is going to ignite it in different ways. For instance, if you have a perturbation that is affecting every species exactly in the same way, it's actually not going to ignite anything. It's just going to make a shift.
Yeah.
And so all the complexity of the dynamics is not really going to be revealed because the perturbation was uniform.
Yeah.
But if you have a heterogeneous perturbation, then it's going to ignite indirect effects between species and so on.
Yeah.
And this framework is, for me, is a convenience framework to think about that. So you're going to take, if you want to say it mathematically, you take your vector of dynamical contribution, the phi dins, so one per species. And then on the other, you have the vector of perturbation. So it's how each species is affected. And you just take the scalar product between the two. That gives you the overall response.
Right.
And so this scalar product can be more or less simple depending on what I'm projecting onto it.
Yeah.
So depending on the perturbation. But to answer and also to say about what mechanistic model you could have underneath, it was at first when I started working on this, I was quite interested in a model where there was some direct biotic interactions like competition by limiting similarity and also niche construction, so transformation of the abiotic environment because I was interested in having a framework where the notion of response and effect trait was mechanistic, and even the notion of function was also a bit more how to say ontological, like the function really had something to do with the system, not just an outside observable. Like you can measure total biomass, but maybe total biomass has no particular pertinence for the system. But if you're thinking of functioning as transformation of abiotic variables that relate to species through their response rates, then it has a lot more, I think, causal interpretation. And so that's why this theory is built to apply to this kind of general model of communities. Formally, I usually think of steady state because then it's simpler, but I think it could be extended out of steady state as well. I mean, you didn't ask that, but I'm just answering it.
No, it's good to hear though. It's good to hear it's more general. That's great. Cool. Okay, I don't see any more hands up. There was a greeting from Danny or a farewell from Danny Montoya in the chat, but no other questions there. So I think I'll close the session. I'll thank Jeff again. It was a really interesting, really inspiring talk. Inspired some great questions afterwards, I think. So thank you very much. And I'm going to stop recording now. I hope this has worked. But yeah, join us again in about a month for the third